Usually when you think about your body, there is this underlying assumption of unity. You look in the mirror and you just see one individual, like one unified self moving through the world.
Right. I mean, it's really the only way we can process our existence on a day-to-day basis.
But if you zoom in, past the skin, past the tissues, down into the cells, that unified self totally shatters. Because according to a concept proposed way back in 1860 by a scientist named Rudolf Virchow, you were actually a Zelenstat, a cell state.
Yeah, a sprawling microscopic metropolis where every single cell is an individual citizen.
Exactly. And a staggering new 2026 review from Nature Cell Biology takes this idea even further. Inside each of those cellular citizens are tiny, specialized organs called organelles.
Which is fascinating because historically biology viewed organelles, things like the lysosome, the mitochondria, as completely independent workhorses, like one made energy, another cleaned up trash, and they just minded their own business.
Right. But our mission today in this deep dive is to explore how these microscopic structures aren't just dumb, isolated factories. They are actually talking, negotiating, and literally running the city of you. We're going to unpack the exact cellular mechanisms of why we age and how your lifestyle actually alters their behavior.
The shift in perspective here is profound. Decades of research prove they are dynamic signaling hubs. I mean, aging isn't just parts wearing out. It's what happens when these hubs lose the ability to communicate.
So to understand the city, we have to meet the key players. Let's start with the cellular recycling center, the lysosome. I've always pictured this as like a microscopic stomach or a cellular trash bag. How does it actually work?
Well, to really visualize the lysosome, you have to imagine a tightly sealed, highly acidic bubble. And the engine driving that acidity is a specialized molecular pump embedded right in its outer wall.
Okay.
It's called the VAT-PACE complex. Think of it as a heavy-duty water pump, but instead of moving water, it uses your cell's energy, ATP, to aggressively pump individual protons from the outside of the bubble to the inside.
And because protons carry a positive charge, I guess packing them into a tight space drops the pH, like it fills the balloon with acid.
Exactly. That intense acidic bath is non-negotiable. Because floating inside that bubble are specialized enzymes called hydrolases. You can think of them as cellular scissors.
Cellular scissors, I like that.
Yeah, and they only wake up and start cutting when the environment is extremely acidic. So when a piece of cellular garbage, like a misfolded protein, gets dropped into the lysosome, those scissors chop it up into raw materials so the cell can build new things.
That sounds incredibly efficient, but also wildly dangerous. I mean, a biological bag full of acid and tiny scissors floating around inside your cells. So what goes wrong as we get older?
The primary culprit is oxidative stress. As our cells generate energy, they naturally produce highly reactive molecules called reactive oxygen species, or ROS.
Right, free radicals.
Yeah, and over time, these act a lot like microscopic rust. They specifically oxidize the delicate lipid bilayers that make up the lysosome's outer wall. As that rust accumulates, the tightly sealed trash bag begins to degrade. It becomes porous.
Oh, wow. So the acid starts leaking out into the rest of the cell.
Two things happen simultaneously, and both are bad. First, because protons leak out, the inside of the lysosome loses its acidity. The molecular scissors go to sleep.
The recycling plant shuts down.
Exactly. Toxic garbage piles up. Second, those acidic hydrolases eventually leak into the main body of the cell, where they can randomly damage healthy structures.
That structural damage really hits home when you look at the Alzheimer's research mentioned in the source. They looked at neurons in mice with Alzheimer's disease, and they found that the lysosomes in those neurons showed constitutive leakage. The bags were completely compromised.
Yeah, it's a huge finding.
But the truly wild part is that when the researchers successfully patched those microscopic leaks, it actually mitigated the Alzheimer's-related amyloid beta aggregation.
It completely reframes neurodegeneration. But what's truly remarkable is how a cell patches those microscopic leaks. It relies on the cell's internal emergency response.
Wait, how does a cell actually patch a microscopic tear in a lipid balloon? There's no biological duct tape.
Well, there's a couple pathways. First, there are these ESERT proteins. They get recruited by a molecule called GABARAP to literally pull the damaged membrane inward, a process called microlithophagy.
Okay, so it pulls the damage inside.
Right, but there's an even more dramatic mechanism called the PITT pathway. When a lysosome ruptures, calcium instantly spills out. The cell detects the calcium leak and treats it like an alarm bell.
A chemical fire alarm.
Yeah, that alarm triggers an enzyme called PI4K2A to make a specific lipid called PI4P right on the torn membrane. That lipid acts as a homing beacon.
So the beacon is set. Who responds to it?
A protein called TECPR1 sees the beacon, swoops in, and grabs a motor protein called KIF1A. Yeah, and KIF1A is basically a microscopic tow truck. It physically walks along the cell's scaffolding, grabs the torn membrane tubules, and literally pulls them out of the lysosome to remove the damage.
So it tows the broken piece away so the healthy membrane can seal back up. That is incredible. Let's do a quick recap here. The lysosome is our acidic recycling bag, but aging and rust make it leak, which shuts down recycling and causes disease. Think about your own cells right now as you listen to this. Are your cellular recycling bags keeping their seals tight, or are they starting to spill into the kitchen?
That's a great way to put it.
And those microscopic tow trucks need energy to operate, which brings us perfectly to the next major player in the cell. The powerhouse, the mitochondria.
Right. Most people remember mitochondria from high school biology. They use the electron transport chain and the TCA cycle to make ATP.
The energy currency.
Exactly. But as we age, oxidative phosphorylation drops, and mitochondrial ROS or mTROS production spikes.
Wait, let's unpack this. We're always told that free radicals and ROS are the villains of aging. I mean, shouldn't we just flood our bodies with antioxidants to stop this rust?
It's a very logical assumption, but biology throws a curveball here called hormesis.
Hormesis.
Yeah, the concept that a mild biological stressor actually makes an organism stronger. If your mitochondria produce massive amounts of ROS, yes, that's bad. But at low levels, mTROS are actually essential signaling molecules.
So they're a cry for help.
Precisely. They trigger factors like HSF1, JNK, and NRF2 to tell the nucleus to build better stress defenses. If you artificially flood your system with high-dose antioxidants, you mute that cry for help.
The nucleus never gets the memo.
Right. And there is an even more brilliant mechanism for when mitochondria get really stressed, called the mitochondrial unfolded protein response, or UPRMT. It relies on a protein called ATFS1.
Let's walk through how this checkpoint works because it's fascinating.
Think of ATFS1 as a corporate security guard whose only job is to walk into the CEO's office. But normally, the guard goes to the front door of the healthy mitochondria, goes inside, and is instantly destroyed by the internal machinery.
So if everything is fine, the messenger just gets tossed in the incinerator. The CEO never hears from them.
Right. But if the mitochondria are stressed, import fails. The badge reader is broken. ATFS1 gets stuck outside, travels to the nucleus, pairs with a protein called DVE1, and turns on massive rescue genes.
If I can't get in the front door, the building is on fire, so I'm pulling the fire alarm. Unbelievable. And while this is happening, the mitochondria are also physically shifting their shape. I want you to imagine a lava lamp. Globs of wax constantly stretching out, snapping into smaller pieces, and then merging back together.
That shape-shifting is mitochondrial dynamics. They pull apart, which is fission, managed by the DRP1 protein, and they fuse together via MFN1, MFN2, and OP1 proteins.
Fission and fusion. What is the point of the lava lamp dance?
To dilute damage. If a mitochondrion takes a hit, it physically fuses with a healthy neighbor. They mix resources, then split apart, intentionally sequestering the damaged parts to be destroyed.
So we have the recycling center and the powerhouse. That takes us to the cell's vast manufacturing center, the endoplasmic reticulum, or ER.
The ER is a massive folded factory where proteins are shaped and calcium is stored. And just like mitochondria, the ER has its own stress response, the UPR-ER. It's monitored by 3 distinct sensors, IRE1, PERK, and ATF6.
The review outlines this really cool analogy of a fire drill. They took C. elegans worms and treated them with a low dose of a toxin called tunicamycin. This caused mild ER stress.
That's a tiny bit.
Yeah, and the result? It triggered a helpful response that actually extended their lifespan. A tiny fire drill prepares the factory. But chronic activation, a never-ending fire alarm, drives diseases like diabetes and cancer.
Which perfectly illustrates why balance is key.
So let's do another quick recap. We've got the lysosome, the recycling center, the mitochondria, the powerhouse, and the ER, the folding factory. But we've looked at these in isolation. You, the listener, don't operate in isolation. So how do these cellular factories talk to each other?
They build physical bridges called membrane contact sites. The organelles actually gather and touch. For example, mito-ER contacts.
Mitochondria and the ER touching.
Yeah, it's where the fission and fusion machinery gathers. It's also a calcium highway. A receptor on the ER, called IP3R, hands calcium to a chaperone called GRP75.
Okay.
And GRP75 hands it directly to VDEC1 and MCU channels on the mitochondria. It's a microscopic bucket brigade for calcium. Interestingly, lowering this specific connection actually delays cellular senescence.
And it's not just the ER and mitochondria. Mitochondria also dock with the lysosome, managed by a protein called Rab7. They physically connect to swap cholesterol and amino acids.
Yes, and here is the domino effect. If the lysosome's pH gets messed up, it deprives the cell of iron. And without iron, it instantly cripples the mitochondria. One organelle's failure guarantees the downfall of the other.
They are completely interdependent, but here is where it gets really interesting. Organelles don't just talk within one cell, they do long-distance calls across the entire body. Tissue-to-tissue signaling. What does a call look like between, say, your gut and your brain?
It's staggering. When lysosomes in a worm's intestine break down fats, a process called lipolysis, they create signaling lipids like OEA and DGLA.
So VIP chemical messengers.
Right. And a chaperone protein called LBP3 carries these lipids out of the gut all the way to the brain. Once there, they activate a receptor called NHR49.
And what does that do?
Tells the neurons to release longevity neuropeptides, specifically NLP11, back into the body.
Wait, a fat-burning event in the gut directly commands the brain to initiate a longevity protocol.
Yes. And it works in reverse, too. Brain to gut. Mitochondrial stress in a neuron releases a Wnt ligand called EGL20. It travels all the way to the intestine and activates the UPR-MT stress response there. The brain panics, and the stomach puts on its armor.
That is just wild to think about. But we aren't alone in our bodies. We have trillions of bacteria in our gut. How does our microbiome factor into these long-distance calls?
Microbial metabolites directly dictate host organelle behavior. Gut bacteria produce chalanic acid via the Kopp's operon and short-chain fatty acids like butyrate and acetate. These actually boost mitochondrial biogenesis in your cells.
So the bacteria are turning the dials on our internal powerhouses. There's also that mind-bending example with vitamin B12.
Oh, right. If bacteria produce low levels of vitamin B12, it lowers a compound called mitochondrial GTP in the host.
And what happens when GTP drops?
The host's mitochondria physically spread out. And that simple spreading extends the reproductive lifespan in worms. The bacteria are literally holding the puppet strings.
That is profound. We think of diet as feeding ourselves, but we're providing raw materials for a bacterial pharmacy. And if cross-body communication wasn't wild enough, the review highlights signals that can actually time travel.
Yes, epigenetic memory and transgenerational effects.
This is probably the craziest fact from the paper. Mitochondrial stress in worms can extend the lifespan of their descendants for over 50 generations. I mean, 50 generations. How is a memory of stress physically passed down that far?
It comes down to elevating mtDNA in the germline. Let's look at starvation. When an organism starves, intestinal lysosomes recruit an energy-sensing enzyme called AMPK.
Okay, AMPK.
That turns on a factor called SKN1, which creates a very specific histone variant called H3.3.
So it manufactures a custom protein tag.
Right, and this H3.3 travels out of the gut and into the germline. Once there, it leads to a specific epigenetic mark, H3K79 methylation, via an enzyme called DOT1.3.
So it's placing a physical bookmark in the genetic code of the offspring.
A permanent bookmark. It passes the longevity and resilience traits to the offspring before they're even born.
So let's summarize this with our third recap. We have fat taxis traveling to the brain, want ligands warning the gut, and starvation formatting the biological hard drives of your great-great-grandchildren. What you are doing right now isn't just changing your cells, it's changing future generations. So what can we actually do about it? How do daily lifestyle choices map onto these mechanisms?
Let's start with dietary restriction and fasting. At a molecular level, fasting lowers a growth pathway called mTORC1 and raises AMPK.
And how does that affect the organelles?
It physically changes the shape of lysosomes. Instead of isolated bubbles, they form a massive, interconnected tubular network. We see this heavily in long-lived E2 mutant worms. If they don't form those tubes, lifespan extension doesn't happen.
So fasting turns individual trash cans into a high-speed recycling superhighway. What about pharmacological interventions? You hear so much buzz around drugs like rapamycin and metformin. Are they acting on these networks?
Absolutely. Rapamycin inhibits mTORC1, which allows a transcription factor called TFEB to enter the nucleus and build more lysosomes. You are chemically forcing the cell to upgrade its recycling plants.
And metformin.
Metformin activates AMPK. It does this either by dropping ATP levels or, amazingly, by binding directly to a protein called PEN2 on the lysosomal VATPase pump.
Wait, it literally docks onto the microscopic acid pump we talked about earlier.
Yes, exactly. By binding to the pump, it kicks off the survival and resilience pathways.
That is wild. And finally, we have to talk about exercise, the most universally recommended anti-aging intervention. What is a jog doing to the Zelenstat?
Exercise drops lysosomal pH, making it more acidic and efficient. It also triggers mitophagy, which clears out weak mitochondria, and as the healthy ones ramp up, they release hormones like irisin and a mitochondrial-encoded peptide called MOTSC.
MOTSC, right.
Yeah, these travel through the blood to physically protect the liver and the brain. Exercise, the ultimate systemic optimizer.
We have covered an immense landscape today. We started with the realization that aging isn't just a generic wearing down of parts. It's a highly orchestrated process dictated by the pH of your lysosomes, the lava lamp splitting of your mitochondria, and the calcium channels of your ER.
And stepping back from the microscopic details, it leaves us with a really provocative thought. We spend billions trying to invent single molecules to fix single organs.
Right.
But if our body is truly a cell state where every organelle relies on constant multi-directional crosstalk, maybe aging isn't fundamentally a disease of damage.
Maybe it's a disease of isolation.
Exactly. Are age-related diseases simply what happens when our organelles forget how to talk to each other? And if so, how can we help them find their voices again?
That is the paradigm-shifting question I want you, the listener, to ponder as you go about your day. Every time you move, every time you eat, you are sending a message down into the metropolis. Make sure you're giving your cellular citizens something good to say.